we have two speakers now, more on the genomics application area. The first one is Nancy Zhang. So she is a collaborator with Peter, and she will say a bit more about the connection you have with Peter. So the floor is yours.
Yeah, thank you everyone for being here and giving me this opportunity. My talk slides are a little different from what is in the program. In the program, it says DNA copy number from bulk tissue to single cells. I decided to take a broader view, and it's genome characterization from bulk tissue to the single cells. So actually, my journey in large scale genomics started with Peter. I remember it was in, I think, 2005. I'm not sure if Peter, if you remember this yourself. Even before I was a postdoc, you were giving a talk at Stanford, and you were talking about the Encode project. And then the year afterwards, actually this fall of 2005, I came to Berkeley and we started working in genomics together. So here is a sort of a timeline. Sorry for the historical inaccuracy. There's a picture of Peter giving a talk. Let's pretend that is in room 200 in Sequoia Hall. So what preceded this was around the time when I was graduating from undergraduate in around 2000, the first rough draft of the human genome sequence was published. And that was a big thing that had a big impact on my outlook towards what I wanted to do. I remember it was you know, I think it was Bill Clinton at that time and Tony Blair they had made a joint appearance announcing that this momentous milestone has been made, that the human genome sequence, the 3 billion base pairs, was known. And actually it wasn't 3 billion base pairs. It was actually not, the last chromosome was not completed until 6 years later in around 2006. But what comes next? What do we do now that we know the genome sequence? Well, we only knew about 1% of it, which is the genes. We knew where most of the protein coding genes were, but we didn't really know what else was in the genome. It was pretty much a dark space. And so in around 2003, what's called the Encode project was launched. And Encode stands for the Encyclopedia of DNA Elements in the Genome. So basically it was an effort to interpret what else was in there, right? What are in the non-coding regions? What are the regulatory sequences that determine when genes got turned on or off? So, you know, so in 2005, I remember Peter gave a talk on Encode. I thought it was really interesting. I had a bunch of questions on that. When I came here for a postdoc, I was a postdoc with Terry Speed, but I talked to Peter about it and he was very welcoming. He welcomed me to the meetings, and that was how our collaboration started. So I want to spend the first 10 minutes just talking a bit about what Peter and I did. To appreciate the Encode project, we have to think about what do we mean when we say we want to interpret the genome? So the DNA, right, is 3 billion bases, and that's organized in 23 pairs of chromosomes. And the DNA sequence is a very long sequence. In fact, it was estimated that if you were able to stretch out our DNA into a very long straight strand, it would stretch 2 meters long. But our nucleus of an average cell is only 5 microns long. 5 microns is 5 millionth of a millimeter. So think about the packing of that 2 meter long sequence into a 5 micron size nucleus. That is a very hard task for any cell to do. And so the DNA is packed in a very regular and very tight and closely regulated way. And that is what you see here. If you take a chromosome, you know, the DNA, that this blue strand is a DNA, and it is it is wound around these things called nucleosomes, and each nucleosome is an octamer of histones. And then these nucleosomes had these long molecular tails that stretch out. These molecular tails can be altered chemically, and that will determine which parts of the DNA is active and which parts are closely folded in and therefore inaccessible. So after we knew the actual basis of the DNA, that enabled these high throughput, large scale profiling experiments to measure what is going on that DNA strand. So that is what Encode is about. In fact, in 2005, when I started working with Peter, it was what's called a pilot phase of the Encode project, where the goal was not to interpret the whole genome, only 1% of it. Because at that time, the biological experiments were very, very expensive. So 1% being 30 megabases. And then what the Encode says is that, okay, there are 35 well-established laboratories in the world that had their own experiments. They could measure different things. Some laboratories measured the accessibility of the DNA. So which parts of this encyclopedia are open and ready for reading into RNA? Which parts are being made into RNA? Because we knew that DNA has to be made into RNA, which then becomes protein. So in order for the DNA to be active and doing something, it has to become RNA. But we know that there were genes that were coded into RNA, but there might be other things. And actually, one of the main things that was found through this project is that most of our genome, even the parts that don't code for genes, are transcribed into RNA, which was very fascinating. And also some experiments measured where do these regulatory proteins bind on the DNA. So in the end, from these 35 different laboratories, about 200 features are measured on the genome. And it's very hard to visualize 200 features, but here is what we always use to, what many people use to look at the human genome. It's called a UCSC Genome Browser. And what it is that, to appreciate, so here is the chromosome. And what is shown here is what's going on in that small segment that is highlighted by a red rectangle. So if you blow that up, that's, this is about 100 kilobases. And then you can see what's going on. For example, these blue lines with the tick marks, those are the genes. And then these small tick marks here is called Encode candidate cis regulatory elements. Okay, so these are cis regulatory elements means like if you think of the DNA as encoding a book and the genes are the nouns of what's being made, then there are these adjectives and verbs that tell you when and how much of this gene should be made. So these are the cis regulatory elements where transcriptional factors bind and other regulatory factors. And then, for example, I just opened up a few of these tracks. Each track shows you a feature. Like this track is telling you about histone acetylation, and that is one type of experiment they do to figure out which parts are active, and you see these peaks here. Okay, so in the end, 200 features were measured, and the pervasive question that was facing scientists is how do the features relate to each other? Okay, does histone acetylation relate to gene activation? Does RNA transcription relate to evolutionary constraint? There were many questions, such kind of questions. And then, so what were people doing to answer this question? They were basically doing random reshuffling. So what is random reshuffling? This is a gross simplification of the problem, but if you think of these two features as 2 point processes, X and Y, the question is how often do they co-occur? And then if you randomly resample Y, say, do a uniform resampling and then compute overlap and then do this many times, that could give you a null distribution for assessing how significant the observed overlap is. So what is wrong with this? So that was where Peter and my conversation started. what was wrong with this is that the genome is highly structured. Okay, genomic features are not uniformly distributed. They're very clumpy, and the genome itself is a highly non-stationary. It's heterogeneous. Okay, so what people were doing, which was this random reshuffling, just gave them p-values that were too optimistic. So then Peter and I just started asking the question, two features overlap more than random chance. Well, what do we mean by random? Because the problem is we only observe one version of the genome that we all have. We only see one endpoint of evolution. So this is what Peter wrote in the paper we eventually published. The essential challenge in the statistical formulation of this problem is the appropriate modeling of the randomness of the genome, since we observe only one. We grappled with this problem, and this was a collaboration with Ben Brown, who will be giving a talk this afternoon, Hai Yan, of course, in the audience, and Nathan Boley. And in the end, I think, I didn't really do anything with non-parametric statistics before, and I really learned this from Peter. And Peter guided us towards a non-parametric approach to this problem, because I remember, we looked at Hawk's processes, we looked at many different, autocorrelative processes. In the end, nothing really seemed to be realistic, because we have no idea what is a random process that governs these different types of features. So, this was actually, I thought, a very, very neat idea that Peter had. Let's say we have two features, blue and yellow, along the genome, and we want to know, we observe that overlap, is that significant? So, so what we did was we took two random windows from the genome that are close together, took only those two windows, and then we basically did a swapping of features. So we took one window, took the blue features from that window, and matched it up with the yellow features from the other window, and vice versa. And then compute the overlap and do this many, many times. And that seemed to us to be a reasonable thing to do. And then so in the end, we had a paper on this where we formulated this piecewise stationary model for the genome. And under this model, we could do, say, stationary block sampling to get standard errors. And also we could do this procedure, and we have normal approximations, the null distribution of overlap test statistics under this procedure. So, and that, what was more satisfying to me than the theory was that in 2007, that was when this initial pilot Encode project went into publication. They used this method called genome structure correction, which is basically that method that started with these discussions with Peter. And that provided p-values for the test of overlap in that paper. And I think, I'm not sure how the biologists, took that, because many of the things that looked significant before no longer were significant. So I'm not sure if it was a very popular method, but it felt to us to be the right thing to do. So now, that was 2007. Where are we now in genomics after 15 years? So incidentally, in April 1st, it was announced that the human genome sequence is finally complete. And that's not an April Fool's joke, even though it was announced to be complete 20 years ago by the politicians. But here is a cover story in Science. That was April 1st. And then what was said was that, because when they said it was complete, it was not actually complete, because there were many regions of the genome that were really hard to sequence because they were very, very repetitive. Repetitive being this like 80 for like thousands of times. And these kind of repetitive sequences are very hard to get right using short read sequencing. And now with long read sequencing, they were actually able to fill in those gaps. But what's more is that during the last 10 years, I think where biology is moving is towards single cell tissue level characterizations and multi-omic profiling. So that is what the rest of this talk is about. And single cell RNA sequencing really started in 2009, but it really took a decade to mature. So for the rest of this talk, I will be telling you about two very recent works. The first is, and now we're starting to get to the copy number stuff. The first is single cell allele-specific copy number, and that came out last year. And the second is on spatial transcriptomic data, which I think is also very exciting. Now, I know that for this talk, you'll see very little mathematics. And there's one technical slide that I might actually have to skip over because of time. But what I'm hoping is that you can see the promise of this data and the many statistical challenges that lay ahead. So both of these papers are on cancer. So I want to first motivate you why cancer is such an interesting system to study. We're going to be doing single cell, okay, single cell studies, which means that for every cell in a tissue, we're going to be doing high throughput genome level profiling. So we will have large matrices where each row is a cell and each column is some feature like a gene expression. Now, when you're looking at single cell studies, what you're really trying to get at is intra-tissue heterogeneity. So what is different between the cells in our tissues? Now, in terms of intra-tissue heterogeneity, nothing is more exciting than cancer, because cancer, I mean, when I think about biology, biology is very hard to model systematically because it's not by intelligent design. Every rule, every model you propose, there is like a million different exceptions. The only systematic way I can think of to understand biology is evolution. That is how biological systems have been traditionally modeled on a mathematical level. But evolution is very hard to study rigorously, even now, because we only see one endpoint of evolution. But cancer is our evolution of our cells, because we are multicellular organisms. Our cells have been trained by evolution to cooperate and form tissues to allow the organism to have a healthy life. But that rule is violated in cancer, because in cancer, what happens is that individual cells become selfish. They gain through mutations selective advantage and adapt to proliferate in tissues and not die. So cancer is a system where we can see evolution happening real time, but you can only do that if you look at things at the single cell level. So, I take it back. In our other tissues during development, there is also selection and adaptation, cell competition and cooperation. That happens throughout developmental biology, but the signals are much more subtle. In cancer, the signals are very strong, which is why we want to look at cancer before we want to look at other things. So here is so what I'm showing you, here is a microscope slide of a breast cancer. And these are our ducts. So this is a ductal carcinoma. And these, you can't see it very well here, but in the ducts are the cancer cells, and surrounding it are stromal cells and immune cells. And one thing that we want to understand is where are the mutations and how is this population of cells evolving within our tissue? And I will show you methods where we basically look at this data. We basically measure RNA expression at the single cell, single spot level. And then we can take this data and find different subpopulations of cells, different subpopulations of or clones of the cancer. So that comes later. So cancer cells follow Darwinian evolution. Okay, what that means is that individual cells gain mutations and they proliferate and they have progeny. The progenies are marked by their mutations. And this fundamentally makes cancer different from other diseases because it changes so fast. For example, if you have heart disease, right? If you have atherosclerosis, you get a stent, you get a bypass, it's done. The cells don't mutate to try to go around your solution. But with cancer, what happens is if you have chemotherapy, radiation therapy, you kill most of the cells, but there's oftentimes a small subclone that emerges that is immune to that. Okay, so that's what happens to lead to metastases. So some cancers mutate fast some cancers mutate slow. The cancers that mutate fast are more clonally heterogeneous. And that is what makes them difficult to treat. So even before single cell came along, through bulk level studies, we find that the clonal diversity, so they were able to use computational methods to get a sense of what is the subclone diversity of a cancer, which is very difficult to do, but there were methods to do that, inaccurate methods. And we were able to find that those cancers with more than 2 subclones, the patients fared worse in the clinic than those with less than 2 subclones. So it was important to look at heterogeneity, and we knew that even before single cell technologies matured. So for example, there's these methods called for multi-region sequencing, which is basically taking multiple slices of the same tumor, sequencing each to get a profile of the mutations in each. And then my group worked on deconvolution methods to take those mutation profiles from the slices and reconstruct the evolutionary tree from that. And you can imagine it's very difficult because we don't have that many slices and these mutations are very, very noisy. So you can imagine how excited we were in 2011 when this paper took basically, I think, about 100 different nuclei from the tumor and sequenced the DNA from the nuclei to get these copy number profiles. That allowed us a first glimpse of the evolution that was happening within the cancer. So during the last 10 years, there were different methods developed for looking at these single cell DNA sequencing data. And I'm going to be talking about this last method that's listed here called Alleloscope, which looks at allele-specific copy number analysis. So before going into the method, I have to tell you about what is allele-specific method estimation. So in our genome, that we actually have 2 copies of our genome, right? One from the father, one from the mother, right? We're diploid, so 2 copies, right? 23 pairs of the chromosome. So so we can, where in places where our mother and father differ in their genome sequence, that's called heterozygote. And then we can just generically call the 2 alleles A and B. So if you have a copy number change, right? So copy number change is when a segment of our genome is amplified or deleted. And that happens very commonly in cancer. That's one of the major things that, that drives cancer evolution. So when you have an amplification, it could have been the blue copy or the red copy, right? We don't know that unless we look at allele-specific signals. When you have a deletion, it could be either the blue or the red that got affected, right? So below, I have the total copy number and the B allele frequency. The B allele frequency just says what is the ratio of the B allele versus the total. And if it's an amplification, 1 copy amplification is 1/3. If it's a 2 copy, if it's on the amplified, or sorry, if it's on the normal copy, right? If it's on the amplified copy, it's 2/3. Same thing with the deletion, depends on whether the B allele is amplified or deleted, it's on the deleted copy, right? So it's either 0 or 1. So one reason why we care about allele-specific copy number is because oftentimes we would have 1 copy deleted and replaced by the other copy. Now, why is that interesting? Because you still end up with 2 copies. It's because each of us here actually inherit lots of deleterious mutations, meaning mutations that would make us sick. The only reason we're not sick is because we inherit 2 copies of our genome. The other healthy copy kicks in. So if you have a loss of heterozygosity, meaning you have 2 of the same haplotypes, then it could be pretty bad. Okay, so that actually happens a lot of times in cancer. Also, so that's, so you have these loss of function mutations that become the primary driving force of the cancer. The other scenario is where you have a gain of function mutation. So a mutation that makes the protein product do something to drive the selection of the cell, increase the fitness of the cell. That oftentimes get amplified during cancer. So by looking at which allele is amplified, you can tell which ones are actually doing the biological, it's a causal factor. So before single cell, right, what preceded our study was that there were bulk studies where they took this tumor, took 4 different regions, and then sequenced it. Here's region 1, here's region 2. And what they did was, this is normal, so there's 2 copies, and they found that there's an amplification. So there's this 3 copies. But what they found is that in these 2 regions, they actually amplified different haplotypes. So this would not have been visible if you were not to look at allele-specific copy number. It would not have been visible even if you were to doing single cell sequencing. And what's interesting is that, what they found later for this tumor is that one of those subclones amplified and became the driving subclone. So, that was the only study we could find of this kind of what's called mirror phenomenon in the bulk setting. And then in 2021, there was this paper that proposed a method to do allele and haplotype specific copy number in single cells. And they found 2 instances of these mirrored subclones in a breast cancer sample. And then their method was applicable to high coverage single cell DNA sequencing data, but that didn't work because most of the data that's out there are not so high coverage. High coverage meaning that there's not as much sparsity, meaning that each cell is covered by a lot of reads, so you don't have that many zeros, okay? So we didn't really know how common these kind of mirrored settings are. So before I tell you about roughly what we did, I'm just gonna reiterate again, what is the underlying structure of the data, okay? So here's a cartoon, right? Here is the normal cell. Normal cells have 2 copies, each one being having a copy number of 1. And then let's say that you have 1 clone in the tumor that has the blue copy swapped for red. So this is what's called a copy neutral loss of heterozygosity. Okay, and then we have another subclone that didn't carry that loss of heterozygosity, but it carried this amplification of this red haplotype. Okay, and there could be many other subclones, but let's just list these 2. But we don't see this. We're not privy to this knowledge. What's observed are very sparsely sequenced reads for individual cells. So here's 1 cell, let's say, and these tick marks are the reads. If they overlap with a heterozygous site, then you can actually read off the allele and say whether it's A or B, right? But you don't know which haplotype it comes from because you don't know how the alleles are linked together. For example, here, the A allele is linked to A, is linked to B, is linked to B, is linked to A. They are together like a pearls on a string. You don't know that. You just know that there's heterozygous site here, heterozygous site here. So that's called phasing. Phasing is when you know how the alleles are strung together. So you see many such cells. Okay, now experiments are generating thousands and tens of thousands of such cells. So the challenges are the reads per cell are really sparse. Okay, so about 1 out of every 1,000 base is covered by a read. We don't know where the breakpoints are. By breakpoint, I mean these positions. Okay, and we don't know what the underlying phase is. So we don't know how the different alleles are strung together into haplotypes. So this is probably the only slide where I tell you about the underlying model. So in the end, we proposed this model. So we said for each cell I, we let the reads be sampled according to a Poisson process with background rate mu t, because these experiments don't have a homogeneous background rate. Some regions of the genome are much more easy to precipitate and sequence. Some are very hard. So even if there's no copy number change, mu t will still have large fluctuations. And then these delta IT tells you where the change points are. They are the cell-specific change points where the chromosome breaks. And then they have, this is the rate function, and the tau i's are the change points. And they are shared across cells because the cells are all progeny of the same evolutionary process. And if read Yij overlaps with a heterozygous site, then what we also observe is the allele that is on the read. And that we assume to be a Bernoulli with data. Data is a major haplotype proportion. I'll tell you a bit more about that later. But basically it depends on whether allele B is on the major haplotype or the minor, major being the one with the more copies. So I think it's easier to look at this picture. So the horizontal axis is a coverage. So 1 means that there's no copy number change. 1.5 means it's 3 divided by 2, 4 divided by 2, 1 divided by 2. And then the vertical axis is the major haplotype proportion. So if you have 3 copies, so 1 to 3 or 2 to 3, then you will be either here or here, right? If it's 4 copies, then you're at 1/4, 2/4, and 3/4. So the key here is that you're looking at single cells. So these should be whole copies, right? So they're discrete. So we had a method. I'm going to really skip through the method. I think there's many methodologies here. You can probably propose a better method. But basically, we did pooled segmentation to find the change points. And then for each region, we did a EM estimation to find out what the major haplotype proportion is. And then we just, we plotted the estimates on this plot. So let me just show you some data. I think that is easier to see. So here is a actual single cell data set. And each row is a cell and each column is a genome position. And this is a male. That's why the X chromosome looks like it's deleted, right? So it's only 1 X chromosome. And you see here, the orange shows amplification. So you have the amplification of chromosome 7, chromosome 8, and chromosome 20, right? So this is single cell data. If you have bulk data, you would just see a profile, a horizontal profile. Now let's focus in on one of the regions that is showing an amplification, which is chromosome 7. You do see a 1 copy amplification as expected. So this is these are the normal cells. They have no amplification. And then this is a major subclone. But then you see here this minor subclone that has the other copy amplified. And this is basically one of the few first samples we had laid our hands on. And immediately we jumped into this mirrored subclone scenario. And what's more is that this seems to be the norm rather than the exception. So we looked at this amplified region. And what we found was that it was very surprising. It was 1 major subclone with 1 copy amplification, right? And then we found these small rare subclones that had this haplotype diversity across the cancer. So what this is telling us is that this cancer is mutating much faster than we thought it was. There's much more heterogeneity than what meets the eye if we were just to look at bulk or single cell total copy number analysis. And what's more is that these different subclones are coexisting in the cancer. Usually these evolutions, they drive out minor subclones, but they're coexisting. So either there is some selective advantage or the mutation is so fast that it's just creating these different subclones. And what's more is that even this deletion has this minor subclone. So, and then, okay, we thought, okay, maybe this is just one. We were lucky we found this one sample that seemed to be very fast mutating. We looked across different cancers, colon cancer, gastric cancer, and breast cancer. And what we found is that the level of heterogeneity, subclonal diversity is staggering, that there was just lots of subclones that were pretty clear. I mean, you can compute a p-value, but you can clearly see it by eye that is very, very significant. So once we started seeing this, when you're looking at genomic data, once you you start wondering whether there's a technical artifact because, these experiments are hard to do. There might be some technical things that are creating these subclones. So my collaborator was able to do what's called a linked read sequencing, where basically they took high molecular weight DNA, which is long strands of DNA, and barcode the reads for that, so that you basically can know the haplotypes, the phasing of the data. And remember, the reason why this is a difficult problem is because you don't know how the phasing. You have to estimate that alongside the rest of the parameters. So we put the phase snips into the data to get a re-estimation. And what we found that the bottom row is a re-estimation. It matches very well with what our method gave without the phasing information. So that validates that what we are seeing is real. And we applied this to the other cancer data sets, and it was real. So what we found is that these cancer cells are evolving very fast. and they have much more heterogeneity than what was previously appreciated. So this is where we move in 15 years. The top is a picture of this cancer. If you were to use methods dated 2005, which is bulk DNA sequencing, you will know that where the copy number changes are in this cancer. And if you were to look at it with single cell DNA sequencing, you now can, with our method, be able to see just much more subclones. And with this, we can actually try to get an evolutionary tree that described how this cancer came about. And we also did single cell ATAC sequencing data, which is a different thing that I don't have time to talk about. But I want to give you just 2 minutes to tell you about what's next in this work. Okay, so now we're using spatial transcriptomic data, and I can let me tell you, because I think there's a lot of opportunities in this data for methods development. So this is what you see under a microscope slide. You see the tissue structure, and if you zoom in, you can see the individual cells. And at about now, 65 micron size spots, which is about 6 cells. These are spots, okay? So each cell is actually, each spot is not one cell, it's like 10 to 20 cells. So 6, now it's getting smaller. It's gonna get to 1 cell size pretty soon. And you can measure different genes. So I'm showing you 6 genes, but you can measure all genes in the transcriptome. Okay, so you can have 20,000 such plots. Okay, so you can see how these different genes are spread out spatially. Now remember, we couldn't do this with single cell because in the original technology, because what you had to do was dissociate the cells and then sequence them. You lose the spatial information, but spatial information matters because the cells, some cancer cells might be interacting with some immune cells. Right, the immune cells are trying to kill the cancer cells. So basically what we did was we applied that procedure to these RNA data with more enhancements to find out where the subclones are. So now we can ask a whole bunch of questions like, how do these subclones differ genetically? Are they intermixed or are they localized on the tissue? And how are they interacting with other cells in their immune and stromal microenvironment? Okay, and then finally, in 10 years, hopefully, we hope to get to the question of how are the cells cooperating and competing against each other? Okay, so I think I'll stop here. And yeah, so basically, in the end, for this data, we found 3 different subclones coexisting. So eventually, actually, I want to skip over this. Eventually, I just want to wrap up by telling you about some of the lessons that I felt I learned from Peter, and we sort of discovered best practices to the data that I think still hold true after 15 years. So the most important, I think, step for all of these projects is the initial step of formulating the null hypothesis. This comes done together with a scientist. When you ask a question that's a biological question, we want to know, find a p-value or whatever. We want to know what is the control, what is normal, because that's not always obvious. In the question about how to assess overlap, what is the null hypothesis? So what do you expect when there's no relationship between the features? So that was, I think, we had many hours of discussion about that. And also, biological data does not conform to clean models. So usually, if you use some kind of parametric model, you find a bunch of hits. Oftentimes, they are model violations rather than things that you're interested in. What that means is that non-parametric methods probably would be better if they're computationally feasible. Or you have to be very careful in your parametric method to make sure that you are taking away those artifacts that you're not interested in. And then one thing that Peter was, set an example on when we were doing the Encode was work very closely with domain experts. That means that we start from the very beginning of question formulation. So usually, when I was a student, PhD student, we thought, okay, you start developing the method when you get the data, hopefully in SR data format file. But you can gain a lot by starting very, very early. And this is especially true in this day when the data is so complex that the biologist needs your help in formulating what is feasible as a question to be asked. So I think that is also an opportunity for statistics as a field that we can actually get involved in the science much early on. We have much better chance to drive it. And I just want to acknowledge the students and postdocs and the collaborators at Stanford who helped me with this work.
Thank you very much for a nice presentation. There's time for one question or comment.
Very good talk. So one question, how do you validate the subclones to make sure it's not the artifact in your models?
Yeah, so we do, there's quite many different ways. For example, we could do, this one, each spot gives us a library of RNA library. So we could go back and sequence that using a different approach. Like, for example, we're doing long read sequencing on some of these libraries to see if we can detect point mutations that coincide with the copy number changes.
So that's what you guys are going to do?
We're doing that now.
And there are also different methods you said. Did you compare with other methods? You listed quite a few. Do you get consistent subclones or you get different clones?
Yeah, so there was only one other method that was comparable that was doing, and that was a PET method called CHISO that was published the same year. And they only applicable to high coverage DNA sequencing, meaning that for every cell you get more reads. So we did, with all these methods, you publish it, you have to do a heavy benchmarking. So we benchmarked against them and show that we could do better.
I see, thank you.
Yeah.
Okay, thank you. I also would have lots of comments, but I'll ask or make them after the talk. In view of time, we have to move forward, but let's thank Nancy again for the very nice presentation. Okay, so the next speaker I announced already, and you can just get everything ready, is Ingi Jessica Lee. She was a student of Hyen and Peter. I think this was quite